Hello and welcome to WinXAS Tutorials, where I want to talk about XANES analysis again. This time we will be looking at the analysis of phase mixtures using reference spectra for fitting procedures and principal component or factor analysis. As always, the WinXAS software and the XANES data I will be using in a hands-on session can be obtained from the WinXAS website. Features in the XANES region like pre-edge peaks or edge position can be analyzed to determine geometry or average valence, as we have seen in the previous tutorial. Additionally, however, the whole XANES region can be used as a fingerprint to identify and eventually quantify the composition of mixtures of various chemical phases in a sample. For analyzing a single XANES spectrum, a sum of reference XANES spectra can be fitted to the experimental spectrum. Provided data reduction of both sample and reference spectra were performed carefully, this procedure should result in the composition of the sample. However, this approach has some major drawbacks. First, any large enough number of reference spectra will eventually result in a good agreement between the sum of the spectra and the sample spectrum. This does not necessarily mean that all these phases are indeed present in the sample. Second, the validity of this approach relies on the database of references and the assumption that reference spectra of all chemical phases present in the sample are included. While there is little one can do when analyzing a single spectrum, alternative procedures exist for the analysis of a series of sample spectra. For a series of sample spectra, meaning hopefully more than 2 or 3, procedures exist that allow us to answer both questions, which are how many chemical phases are present in this series of spectra, and are suitable reference spectra as required for quantifying these phases included in the database of reference spectra. The corresponding procedure is called principal component or factor analysis. And in the following, I want to briefly talk about the fundamentals of factor analysis, in particular about how it is implemented in the WinXAS software. Factor analysis has been around for a while, for instance in chemistry as a part of chemometrics. A definition of factor analysis states that it reduces data to their lowest dimensionality, which is the number of chemical phases in our phase mixtures, by means that yield recognizable factors, which is the chemical identity of the phases present in the samples. This seems to be exactly what we need for a reliable quantification. So let's see how this is achieved. Factor analysis is a multi-step procedure that starts with the series of suitable, which means carefully pre-treated, experimental XANES spectra. Next comes a technique known from linear algebra as singular value decomposition, which among other results yields the number of primary factors which correspond to the number of phases in our samples. Thereafter, factor analysis branches out to various approaches used to determine those recognizable real factors that correspond to chemical identity of the phases in our samples. The procedure implemented in WinXAS and used here is referred to as target testing or target transformation. Eventually, the combined information of number of phases and chemical identity of those phases can be used for quantification. Singular value decomposition is used to decompose a matrix that contains the XANES spectra in columns into 3 matrices that contain abstract factors, eigenvalues, and weights. This is the heart of factor analysis, and all that follows uses these matrices to determine the number and the chemical identity of the phases in our mixtures. Note that to this point, this is a model-free approach that has not used any reference spectra or databases. After singular value decomposition, the number of primary components and the border between primary and secondary components is determined. While primary components carry the relevant data, secondary components contain the noise. Various approaches can be used, such as reproducing the spectra from only the primary components and evaluating the difference between original and reproduced spectra. Additionally, certain indicators are available that exhibit a minimum at the border between primary and secondary components. Having determined the number of phases in the samples corresponding to the number of primary components ends the model-free analysis. To convert the abstract factors into real factors and assigning chemical phases to the primary components requires a database with suitable reference spectra. A possible procedure for this task is called target transformation. Here, the reference spectrum is multiplied by 2 matrices containing the primary components in columns or rows. The result of this multiplication is another spectrum that can be compared to the original reference spectrum. A good agreement between the two indicates that this reference is a likely candidate for one of the chemical phases in the mixture, or not. In this manner, all spectra in the database are tested, hopefully resulting in a number of references that correspond to the number of primary components. If this is not the case, quantifying the phase composition of the mixtures in the series of experimental spectra is not possible. Eventually, we are somewhat back to where all this started, with refining a sum of reference spectra to the experimental spectrum and determining its phase composition. However, after factor analysis, we can be much more confident in the results because we know the number and the chemical identity of the references to be used, which in turn confirms the integrity of our database. In theory, the phase composition can be directly calculated from the matrices obtained by the factor analysis procedure. While this is implemented in WinXAS and may work for a near perfect fit between chemical phases and references, XANES refinement remains the more universal analysis approach for a variety of sample spectra. Let's finish this introduction to factor analysis with some remarks or word of caution, if you wish. Factor analysis is a powerful tool for analyzing series of spectra, no doubt about that. However, carefully prepared is key here. It refers to measuring both samples and references during the same beam time under identical conditions and, of course, later using exactly the same data reduction procedures for samples and references. Even then, additional pitfalls may have to be considered. For instance, available references at room temperature may not correspond to the phases forming under reaction conditions at elevated temperatures. Changes to the XANES region with temperature will affect both target testing and quantification. Moreover, a continuously changing temperature during measurement may even result in an additional primary component. Eventually, an invariant ratio of two chemical phases will have them appear as only one primary component, which potentially can entirely ruin target testing and quantification. At this point, I can only recommend keeping all this in mind when analyzing your own XANES data. Coming next is the hands-on session. Objectives will be performing XANES reference fits and factor analysis on a series of vanadium K-edge spectra measured during reduction of vanadium pentoxide. Vanadium sample and reference spectra are already calibrated, normalized, and the sample spectra are contained in a single multiple column file. Showing all these steps on a series of raw data files from an in situ experiment will be the subject of another tutorial. The vanadium oxide XANES spectra used are included in the tutorials data folder, which can be obtained from the WinXAS website. Let's get started and open the experimental data file. In the tutorials data folder, vanadium oxide PCA, we find this file that we can open. This is a multiple column file that contains all the vanadium spectra that we want to use for quantification and for factor analysis. Since this is a multiple column file, we can use some display options available here in the time resolved menu, multiple column plot, and we don't need to change anything here, just OK. And we get all the spectra in the file superimposed in one plot. We do see the characteristic shift in the vanadium K-edge with reduction, the decrease in the height of the pre-edge feature, and we see those characteristic isospastical points that indicate a changing ratio of the same constituent phases during a chemical reaction, for instance, in this case, that we want to analyze. Okay, first thing we are going to start with is the XANES fit, which is using references to perform a simulation of the spectrum and to use that simulation with different phases to perform a quantification of those phases. This is in the XAFS menu. LC XANES fit is what this procedure is called. Since we're going to do this multiple times, I want to write this into a macro so I do not have to select the references and constraints for the parameters every time. I just do this once, write this into a macro, and then I can apply that macro to the spectra that we want to analyze. So this option in WinXAS is available here in the top left corner. There is a start and stop button for the macro recorder, and then there is a play button for this macro. The details are described in the manual, but basically all the
items you find here for data treatment or data reduction or data analysis can be saved into a macro. Multiple steps of those can all be saved in one macro. There is no editing feature for the macro, so if you want to change a step, if you want to remove a step or add a step to an existing macro, this is not going to work. You have to record a new macro. Graphical changes, for instance, opening a new file or changing a graphic window, will not be recorded in the macro. This is basically only for those data reduction and data treatment steps. Okay, we are starting the macro. We need a name. Vanadium XANES. Save. So now this recorder is running, and whatever we do now will be recorded in this macro. If you want to stop it, just press this button again. So what we want to have is the LC XANES fit options, and first thing we need to load the XANES spectra, the reference spectra that we want to use for this simulation and refinement. So we do have in the same folder some references for vanadium 5, 4, and 3 oxide. So we can just select those in any order. If you want to have them in a particular order, side note, the one that should be in front here needs to be selected last. So for instance, if you want to have it in the order of increasing average valence, you would click on V2, VO2, V2O5, and V2O3. And then the last one is in front. Okay, that'll do it for now. Open, and we have selected our references. Now click on OK. So here we need to input the starting parameters for those phases for the refinement. There are two parameters. One is the concentration. The other is an E0 shift that is being applied to the spectrum, to the reference spectrum during refinement. Apparently, the outcome of this refinement is very sensitive on the edge position. So even very small changes in the edge position of your reference spectra might hinder the refinement, might result in the algorithm not actually finding a minimum. And so there is this option to shift the reference spectrum even so slightly on the energy scale and hopefully find a better agreement between theory and experiment in the refinement. This should, of course, be very, very small, and you may even set it to 0 for the starting parameter. Well, because we're doing the refinement now in keV, this is already 1 eV, and this is a lot. So whatever value you see here later in the results should be much smaller than that. Large number for the E0 shift may actually indicate that the references refined is not really present in the sample. It is actually just an artifact of the refinement and not a real reference that should be used. Anyway, since we are using this refinement, this macro that we are currently recording, not only for those cases where all those three phases are present in the samples, but also, let's say, for the last and the first and the last spectrum. So we start with V2O5, where we only have V2O5 present, and we end up with V2O3. So since we want to use the same macro in all cases, we do want to constrain those parameters to avoid them, for instance, with the concentration exceeding 1 or being below 0. So for this, we go to constrain. There's a box down here and constrain, and now we can select borders for those concentrations. So we do want them to vary only in the range between 0 and 1, and we can do this for all of those. Now for the concentration. Those things will also be saved in the macro. Same for the E0 shift. We do want some flexibility here, but not too much. We do not want the spectra to be shifted by 10 or hundreds of EV. So again, select this, go to constrain, and let's assume we just use a range from minus to plus 1 EV. That should be plenty to account for slight differences in energy calibration of the references and calibration of your sample spectra. So we have this here, here, and the same here. So those are our starting parameters, and since there are not that many parameters as starting values, those numbers should suffice. So we just click on OK, and we get a refinement. Can just again on OK, and we do not want that. So this is the agreement. The red trace is the experimental trace. The blue trace is the simulated spectrum. Now we can switch off the macro because this is the only step that we actually wanted to apply in the macro. So we can switch that off now so that we don't accidentally record more steps in the macro than we actually want. We can have a brief look at the results. So there's a concentration and there's the E0 shift, and apparently we start with V2O5 with rather pure vanadium pentoxide in this reduction of vanadium pentoxide to V2O3. So we basically have a concentration of 1 in V2 in this case. Okay, let's select another column, another spectrum from this multiple column file. We can go up here, and there are in total 14 spectra in 15 columns. So we can select something in between. Let's say we go for 10. And now we can repeat this refinement. For this, we do not need to go here and input all the starting parameters, but we can just use our macro now. So we click on the recorder and play the vanadium XANES macro that we just saved on OK. And yeah, again, we get a rather good agreement between the experimental spectrum and the simulated spectrum. In this case, now we can display the individual references that were used for this refinement, and we see that actually all 3 references now were used for simulating the blue trace in varying concentration. The concentration are given here. So yeah, almost all about 30 respective 40% concentration of the different oxides here that were used to simulate or to calculate the blue trace and refine this blue trace to the experimental spectrum at this point in the series of spectra, which is the red trace. So that looks good. If you look at the reference as the E0 shift, those are very small. All this indicates that those references are indeed present and need to be considered to simulate the experimental spectrum. However, as I pointed out in the first part of this tutorial, there is no way to be sure when just refining the sum of spectra to a single experimental spectrum. We do not know how many references we really need. We do not know how many phases are present in our mixture, and we cannot be certain that the references selected are indeed the chemical phases present in the sample spectrum. All looks very decent and all looks very probable, but there is no certainty here. So there is little we can do when analyzing a single spectrum, as I pointed out. But of course, we can improve the situation when we have a series of spectra. Then we can use the factor analysis and try to get a better idea of the number of components, the number of phases of references that we really need, and if those references that we have here are indeed the ones that should be used to simulate the spectrum. And that's the next step then. That is the principal component or factor analysis. However, before we get to factor analysis, maybe a few words about saving data from WinXAS that you may want to import into another software for graphical purposes or for data analysis. If you want to save WinXAS data into a format that can be read by other programs, you need to save it as an ASCII file. So saving as an ASCII file will basically save whatever you see on the screen at this moment. So currently you would get a multiple column ASCII file that contains the experimental spectrum, the simulated spectrum, so the blue trace, and the individual reference traces as shown here. If you switch off the display of the individual reference traces and go to save ASCII file, the program only saves the experimental spectrum and the simulated spectrum. And if you switch that off as well and go to save ASCII file, the program only saves the experimental spectrum. So this is the way this is handled, and this is the way to export data, export results from WinXAS to then import them into some other program. Okay, let's get to factor analysis, and we want to apply the factor analysis to the multiple column file that we are working on and that was created before for that purpose, so we do not have to input individual reference or individual spectra, which is also possible. So we take the columns from the multiple column file and perform the singular value decomposition. So the math basically behind factor analysis is almost done at this point. Well, the program has performed the singular value decomposition, so we have the three matrices that I introduced in the first part of this tutorial, containing the component, the weights, and the eigenvalues. So how to proceed now with whatever we have on the screen at the moment. So first of all, when we start with 14 spectra, we get 14 components. So the question now is, how many of those components are required to reproduce the experimental spectra? This is what this display is for, which currently shows a red and a blue trace. And the red trace is the experimental spectrum and the blue trace is the reconstructed spectrum, the first reconstructed spectrum, as you can see up here. So now we can basically go through the experimental spectra and compare experimental spectrum and reproduce a reconstructed spectrum and see the good agreement. which is not surprising because we are almost using all of the principal components. So now the question is, how many of those components are primary components? How many of those components contain the data and the others then only the noise? So what we can do now is we just select the first component. They are ordered in with decreasing weight. So the first one is always the one with the highest weight. We just select one, we go to reconstruct spectra. And now, of course, we do see a significant difference between the experimental spectrum and the reconstructed first spectrum. And this will be the same now if we scroll through the experimental spectrum, we would always get this difference. Which is not surprising because we are only using one component, which is obviously not enough to reconstruct the experimental data. So now we can continue. We select two components, go to reconstruct spectra, and see that significantly improves the difference between the red and the blue trace, so the difference between the experimental and the reconstructed spectrum. Again, we can scroll through the reconstructed spectrum or spectra, and we see there are still regions where this reconstruction does not work so well. So next step would be then to include three components, reconstruct, and if we now scroll through the experimental and reconstructed spectra, we see a very good agreement between the two traces. So additionally, we can look at the residuals. I will start with that by just using the first component, reconstruct, and show residuals. That shows the difference between the experimental and the reconstructed spectrum. So as a function of the number. The inscription here, ignore the inscriptions. This is basically the difference, the sum over all differences of the data point square as a function of the number of the components. So there's significant difference there, apparently. So now if we select 2 and reconstruct, go show residual, again we see in this point, in this range here, between 8, 9, 10, 11, we see still some significant disagreement between the experimental and the reconstructed spectrum. So only now if we go to 3 components, reconstruct and look at the residual, we basically have little residual left, and this then of course mirrors the good agreement between the experimental and reconstructed spectra over the entire range when we use 3 components for the reconstruction. So this will not significantly improve if we use more components than 3. So this is the first indication that 3 components should be sufficient to reproduce the experimental spectra. What else is there? Well, we can look at the abstract factors themselves. This is the first component, the first abstract factor, the first principal component. This is the second one, this is the third one, and this is the fourth one. And all the others after that, you can clearly see they show mostly noise. So the data indeed is contained within the first 3 principal components. These seem to be the primary component that contain the data. The fourth and higher components only show the noise. The same is visible from the indicator. This is this indicator that I mentioned in the first part of the tutorial that is supposed to go through a minimum at the border between the primary and the secondary components. And it does that. We have 16, 6, 4, 6. So at 3 components, indeed, it seems that those 3 components contain the data, are sufficient to reproduce the experimental spectra within the noise, and the remaining components then only show the noise. So this is our number of primary factors, of primary components. This is the number of references we need now to quantify, to reproduce the experimental spectra really, and to do a quantification at the end. So as I said, this number is basically a model 3 result. So far we have not used any references, we have not used any databases to just get to this result that tells us 3 components are sufficient to reproduce the experimental spectrum. So now next question is, what are those components? How do we get from the abstract factors that are our components to the real factors that are the reference spectra? And as I explained previously, the procedure implemented in the software is called target transformation, and it uses references, calculates another spectrum of sorts, and if the two are a good match, if the two agree, then the result is indicative of using, or that this spectrum, that this reference can be used for quantification. So for this procedure, you need to select the number of primary components, because remember, those are the components used in the matrices and in the transpose of the matrix for this target transformation multiplication. And so those are the primary components. And then we can go to target transformation and select a reference spectrum. Those are the references that we used also in the previous tutorial. So let's start with V2O5, with the vanadium pentoxide. Click on OK. And as we already saw from the quantification, that is definitely one of the references that we need to use for the quantification. So there's a red trace now and there is a blue trace, and the very good agreement between the two really indicates that the vanadium pentoxide reference is a good candidate for one of the chemical phases, in this case vanadium pentoxide, that is present in the mixture of phases that are our experimental spectrum. So we can save this now, we can add this to the list of references that we have identified. So in the same manner, we try out the next reference spectrum, which let's go with the vanadium 4 oxide. And again, we get a good agreement, reasonably good agreement between the red and the blue trace. So VO2 is also one of the references that we probably want to use for quantification later on. So we save that as well, and we are looking for 3 references. We have, fortunately, in the very limited database available, we have 1 more reference, and this is the V2O3. And we can try that. And again, we get a good agreement between the reference spectrum and the target transformed reference spectrum, so the red and the blue trace. And again, we save this reference also to the list of identified references. So fortunately for this case, we were looking for 3 references, 3 primary components that we needed to transform into real factors, and we found in our very limited database 3 references that are based on the target transformation good candidates for quantification. So this is basically, or this is normally the end of the factor analysis. We have determined the number of references necessary for quantification, and we have, based on our database, identified this number of references, so chemical identity species that we can use now for quantifying those mixtures, those phases in each individual spectrum of our series of experimental spectra. So just briefly, because I mentioned that in the tutorial, show real concentration basically now calculates the concentration of those references in the experimental spectra. So this can be done. And if you get a very good agreement, like in this case, a very good agreement between target transform and reference spectrum, then you can indeed calculate the reference, the concentration of those references in the experimental spectrum. So there is no XANES fit required in those cases. Well, this is the end of the principal component analysis. Next step now would be to quantify the concentration in regular cases, quantify the concentration of those phase mixtures in the experimental data by linear combination XANES fit. Some concluding remarks maybe to what we did here and this very good agreement between experiment and theory, if you wish, the very good results that we got from the principal component analysis. By now you may have already guessed that this is constructed data, that I actually used those references to manually construct those experimental spectra as mixtures of those references. So if you wish, what we did so far is a proof of principle. You see that it works, and the concentration we determine here correspond to the concentration of those references in the constructed experimental spectrum. But it's not only proof of principle, it also shows a best case scenario. So this is basically the example where your references, the structure of your reference compounds perfectly match the structure of the chemical phases present in your phase mixtures. So this is possible to obtain in those cases. When your references are too perfect, as I mentioned in the first part, when they are too good actually to be or to really correspond to the mixtures, to the phase mixtures measured, for instance, under reaction conditions, then the agreement that you obtain from factor analysis and also from target transformation will not be as good as shown in this example. Also, there is no additional phase. We only really find 3 primary components, 3 real factors. If that was a real in situ experiment, for instance, a temperature programmed experiment where you study the degree of reduction of vanadium pentoxide as a function of temperature in hydrogen atmosphere, for instance, then you would increase the temperature during the experiment, and this temperature would somehow affect the factor analysis. For instance, actually show up as a 4th component that has no reference, but is just basically due to the increasing temperature during the experiment. So this needs to be kept in mind when actually applying this procedure to real samples and a real example. We will come back to this later in another tutorial when we will look at a real in situ experiment, and then we will talk about this in some more detail. Okay, but so far this is it for the factor analysis. We have the information that we wanted to obtain, the number of components and the chemical identity of the phases present in the phase mixtures. After factor analysis, WinXAS saves all the data that you just saw in this box in the result window, and also in a number of output files that it saves in the corresponding data directory where you have also the experimental spectra. So here you see the indicators, for instance, you see the target transformation R value. So this is the difference between the reference and the target transform spectra, and you see those values are all well below 1%, indicating a very good agreement, as we saw, between reference and the target transformed reference spectrum. Okay, so let's go back to the original data. So those are all the files now generated. Those were not present here before, and those are all the files generated now from the principal component analysis procedure. Well, let's go back to the original file, the series of spectra. And now we want to do the quantification using the XANES refinement that we tried out earlier. We did the principal component analysis now, so we are pretty confident that we need 3 references and that we have those references in our database and that they are the V2O5, VO2, and V2O3 oxides that are present here during this reduction of vanadium pentoxide. So instead of applying the macro that we saved earlier now to every single individual spectrum in the series, we want to use a batch analysis. So we have a multiple column file, and the reason why I actually generated this multiple column file is that it permits this type of batch analysis. So I can apply the macro that we saved earlier now in a batch analysis to all the spectra in the series in this multiple column file. This is done in time-resolved automatic analysis. So automatic analysis, there's a number of boxes here that you do not need. But the automatic analysis basically has 2 types of output, and you need to decide at this point what you are interested in, what you want to save. Basically, the output can be numerical values, so say fit parameters, or it can be traces data curves. So for instance, if you perform the data reduction, so including energy calibration, normalization, background correction, atomic absorption correction, and the Fourier transformation, all in one macro. Then the Fourier transform chi of k, so the radial distribution function, that would be the result that you are interested in at the end and that you want to save. So in this case, you need to save curves. You need to save the resulting radial distribution function. In our case now, we are interested in the fitting parameters. We are interested in the concentration that we obtain from this XANES refinement. We're not so much interested in the experimental spectra because we already have those in a file, but we are interested in the numerical value, the numerical results from the fitting procedure. And so now for this automatic analysis, we need to select the option Save Fit Parameters. Click on OK. And now we select the macro. So you have to have a macro recorded in order to perform this batch analysis, but we have that. We can use the vanadium XANES macro. Click on OK, and that will automatically perform this fitting procedure to all the 14 spectra in the multiple column file, the series of vanadium spectra that we were analyzing here. So this is the last spectrum now. Shown here is the result of the last refinement. So this is basically only V2O3. Yeah, this is the last spectrum. After reduction, the result is V2O3, and this is then basically here 100% V2O3 at the end. So now, where are the numerical results? Where is the concentration saved? The concentration, the output of this batch analysis, of this automatic analysis, is saved in a separate file, which is called autodex parameter. Automatic analysis, the parameters. There is currently no option to change this name, so your fitting parameters will always be saved in this file. And it will be overwritten. So if you want to save the parameters from a previous refinement, you need to rename this file. So now this is what we want to open, and it's a multiple column file, again an ASCII multiple column file, so you can open this also with other software if you like. And it contains the parameters, the fitting parameters. Remember, the parameters that we refined were the concentration and the E0 shift. So what we see now here is the concentration, the evolution of the concentration of the first phase in the refinement, in the macro that we use. So this is V2O3. This is the evolution of the concentration of V2O3 during this reduction. This is in the first or in the second column. This is the first thing that you load or that is displayed when opening this file. So if you wanted to see the next concentration, this is for the fourth column now. The third column would contain the E0 value, which we're not interested in, so we open the fourth column now. So this is again evolution of concentration as a function of spectrum number, just like that. And this apparently corresponds to the vanadium pentoxide. And then we select 6, and then this is the intermediate that is formed. This is the VO2 that is formed during the reduction. And now we can just combine those, and we get the evolution of the phase composition during this reduction of vanadium pentoxide to the V2O3. And the formation of a VO2 intermediate phase here during this reduction. So this is of course very similar to what you already saw from the calculated concentration in the factor analysis dialog box, because as I said, this is constructed data. So basically this procedure reproduces the concentration that I selected when constructing those spectra for the purpose of showing how this procedure works, and of course also for a proof of principle. So we get the same concentration, the same evolution concentration here that we already observed in the factor analysis. So the procedure works. Again, it's a best case scenario in a sense that you have perfect references for that. The reference spectra really correspond to the reference to the phases in your experimental mixture. This will normally not be the case, especially if you measure in situ, operando, spectroscopy under reaction condition at elevated temperatures, temperature programmed, and so on and so forth. Then this will probably be a different picture. Okay, so this is what I wanted to show with respect to factor analysis, to phase analysis from XANES spectra, experimental XANES spectra, using experimental references for the analysis of the phase composition. And this is it for this rather long tutorial, and I sure hope to see you in the next one.